So my question is, how can we, according to your experience, how can we make this happen, that we have a close collaboration? Because it's not so many funding agencies in the field. We still have the ATA, but then I think it's already getting a little bit difficult. How could this be happen? How could this manifest? And the next question, do you also think it would be useful to involve industry? And if yes, at which time or by which level?
Well, that's such a great question. Maybe I'll start and then, Ralph can kind of pick up from his perspective also. So I'll start with the second half of the question, industry. I think we are still trying to figure out what the industry landscape is for tinnitus. It's quite complex, right? I mean, I almost feel like in the dementia space, it's way more straightforward. You've got, major biotech companies, small molecules, ASO therapeutics. I mean, it's a pipeline that makes sense industry-wise. What I think will end up happening in this field as far as engaging industry is you all will probably reach a point where you actually are building a drug discovery pipeline of some kind. And that's when you're going to want to bring in industry, and you can have those in the form of tinnitus roundtables. We have these in other fields where interested companies and even investors are brought together to really discuss, does anything look investable? If we were to run a trial, what would that mean to validate something? So I think funders can help you all make that happen. We can bring the industry partners in the room. And then your first question was, how do we initiate more funders collaborating? Well, it's happening right now. All of us being together for these conversations is the start of a collaboration. And then funders behind the scenes will be reaching out to one another and discussing what each funder's sort of mission is, kind of their goals, and where there's alignment and funders, that alignment point and mission and goals is where we can collaborate, and we can either work together, to design new programs. We could co-fund together. We can create conferences together. But we need, as funders, to come together and find those alignment points because they sort of differ across funders. For example, Rainwater Charitable Foundation, we are not a patient advocacy organization. So we don't work directly with patients, and that's not even our mission. Our mission, quite frankly, is just to develop cures as fast as possible. So we have a kind of a shift into later translational research focus, whereas other funders might be interested in other aspects of supporting the tinnitus skill. So I'll pass it over to Ralph because I want to make sure he weighs in from his perspective also.
So from obviously R&ID, and so we're a sort of UK based funder, but we fund internationally. I think on the industry front, I mean there have been a few sort of biotech companies that have tested drugs in tinnitus, like Autonomy, like Tiffany recently, but they tend to be like small sort of single asset sort of companies. I think our interaction with sort of large pharma is that they're definitely aware of the opportunity, but my sense is they're playing a sort of a watch and waiting game, and so they're kind of waiting for a really good target to come along, and then I think they will kind of invest and take it forward. So I think as charities, funders, I think our role is to try and understand what industry wants, because ultimately they're going to have to commercialize and bring these treatments to market. So we need to make sure that they that we understand what they need. And I think as charities, we're trying to sort of create those opportunities and de-risk the science so that commercial investment can sort of come in. So we have a kind of like an ongoing sort of dialogue, I think, and engagement with companies. I think I'll just echo all of the points around how charities and funders can work together. So R&ID, we've collaborated with the Tinnitus UK, was obviously the British BTA before, so we've co-funded PhD students. So we've sort of combined resources. we've also worked with Alzheimer's Research UK in the UK. Not on the tinnitus space, but I think we can replicate it in trying to kind of focus research effort on understanding the link between dementia and
Hearing this, I think my microphone's covered up. I can't hear anybody.
from focus. I'm sorry, thank those. Start again.
My digital twin.
Yeah, so I was just saying, so we have collaborated with like dementia charities to try and understand the link between dementia and hearing loss. And so again, I think it's great when funders from slightly allied fields have come together, because not only do we kind of leverage and bring our funding together, we can sort of bring new fields together as well. So I'm kind of keen to explore that, and I think there's an opportunity that I'm keen to explore is how, it's building this kind of momentum. Like how do we get like big national funders? So I guess like the NIH in the US, like the Medical Research Council in the UK. So these like really kind of like big funders. How can we get them putting more funding into tinnitus research and hearing research? I think coming together collectively as funders, I think we have a louder voice when we go and talk to those sort of national funders if we're doing that together rather than individually.
Right. Great. Maybe we can hear also from Alex and then you, Shane, just to add your thoughts on the role of funders in taking the field forward.
Yeah. So I'm Alex Bruce-Johnson from Tinnitus UK. I think if you think about the reasons why organizations don't collaborate, I think it helps to understand why they should. I think organizations don't collaborate because egos can play a part in it, especially in smaller organizations where the ego has a bigger influence over the organization. I think that can definitely play a part in the voluntary sector. I've seen it. And also, I think the other reason is it's a skill. Being able to collaborate with other organizations is a learned behavior. It's a skill that people and organizations learn. If you want to do it effectively, if you want to do it on a surface level, superficial level, and for it to not be particularly meaningful, that's really easy to do. But actually to collaborate on a deep, rich set of aligned values, then that's a skill which a lot of small voluntary sector organizations just don't have. So I think that's where sophisticated funders can come in. That's where we can share experiences between small organizations and large organizations and share that learning. But I think if you flip it around, then I think it's, events like this where we can understand what those shared principles are, those shared values, and identify a common purpose. I think they're the ingredients to at least get the conversation started about collaboration.
Awesome. Yeah. Shall we go to Shane first? Because then we've covered sort of the funding organization.
Right. So I would just add, Hearing Health Foundation is very U.S. based. The people that we fund are all in the States. So that is one huge limitation that we have. We did from Julia Campbell. So I'm thrilled to see that she's part of this Thai consortium that you have. But I feel like, I'm very honored to be here because we haven't really done anything in the EU. But I think that's our biggest hang up now is that we don't really do beyond the US. I did talk to Kelly last night about TRI. I feel like that's something that we should explore. The other limitation that we have is that we fund individual researchers. We don't really do institution to institution or foundation to foundation, so we haven't really explored that at all. So that's what's happening with us.
Great. Yeah, I think Bertolt, you had a question. But so in the meantime, guys, we have like 45 minutes still before lunch. So it's open discussion. You can also recall, back points from Dan and Dirk's presentation. I put up some of the main points on the screen here. But
can I have a last one?
Okay, let Sven also respond.
Because I think
and then we got
since you all know I'm not an expert on tinnitus, but I'm pretty much an expert on creating companies and leading them at least to partial success. So before we ask to bring the industry to the table, if we look at the situation in tinnitus and let's imagine that some collaboration, some research institution finds for example, a drug that potentially could lead to a final product, then we have to then setting up the company and bringing it to a point where there is let's say a market intake is actually pretty easy. The hard thing is to get the finance. So the question in that regard is way more not when we get the companies on board, it's when we get the capital on board. So the venture capital that is unfortunately mostly located in the US, not so much in Europe, unfortunately. This is something I think that's very important. So the collaboration on the private organization that fund tinnitus research today will have to make up their minds how to finance findings in the second step, because this is basically what we see in the last 3 months. If we talk to little startup companies who had an idea, is basically they all lack the secondary fund. So that is basically where everything dies. And I think this is something that especially the collaboration of the private institutions really have to give an answer to, because it doesn't matter how great the research is and how great the product is that comes out if we don't get the secondary funding to actually establish this as a final product. And the second thing I think that's lacking to speed up research is often decision-making processes. So a lot of companies in all fields die because their decision-making processes are too slow, too complicated, and not directly in quick. So that's something in the collaboration also of the private funders. That's something super important. We tried to ease that up with the decision-making process. We applied on our research grants to somehow decomplify the research applications and put a more, let's say, business approach and let the people pitch the ideas, because we have a lot of experts sitting there and they're very quickly and smart enough to understand if an idea is something that has an actual basis or doesn't. So that's, I think, a second very important thing. And the international collaboration, that's, I think, one of the most important things, sharing information, because that's something the businesses all around the world do. And this is why developments are there much, much faster than often in science. I understand that there's a whole different world of complexity behind it, but it's something I think that's basically the most important.
All right, thanks. Wait, so I think we still Bertolt, who else? And then Marlies and Winnie. Okay, yeah.
Yeah, I think we have to be aware that we are a small field, and at the end, the people sitting here are more or less similar people who work in the type project, similar people who are active within the TRI and who are sitting on the board of RNID and ATA. So it's all a relatively small community. And for sure, if I look on the field from outside, I would say it makes very much sense that these different organizations try to collaborate as much as possible and do not compete with each other. In other words, for me as a researcher, it makes no difference whether a donor gives money to Tinnitus Quest or to ATA, to RNID, and then they give a grant to our institution. So at the end, I don't care from where it comes from, but it would be very good if these different efforts would be harmonized in a certain way, that also we do not spend lots of time writing 5 different grant applications, reviewing the same grant 5 times for different organizations. So it would be nice if there could be some way of harmonization. For sure, every single association has maybe different constraints. That's fine, and I think also maybe different philosophies, but if there would be some kind of harmonization of the format or transfer of applications, I think that could make our work just a little bit easier. And I think the second question, I agree with you that I think at the first level, it's task of academics, of charities, to bring ideas to a certain level, then venture capital has to kick in, and then at a certain moment you have the established industry. But so my question before was, in order to bring either people from venture capital or from industry in this group, in this discussion, they might provide us with the information what would be needed from their perspective. What is lacking at the moment, and what is the most urgent need for us, and so their task for us, what should we do, what should we achieve in order that they would be considering kicking in.
I just want to add to Bertel's point on the importance of harmonization between the funders, because I think it's really something that the funding organizations in the room should explore further. And maybe that's like a Zoom call we can have at some point in the coming months. But I think it's an important point we need to follow up on. Okay, yes. Winnie and Marlies then after. Yeah.
I wanted to say a quick comment about the last bit, if possible.
Yeah, go ahead.
Okay, so there was a lot discussed, like how do we get venture capital interested in this field? They were not interested in primary topathies either for many years, until we had to do a few things for them before they were like, okay, this is worth investing in. Number one, they didn't know where to find patients. There needs to be a centralized, accessible point where a company that wants to start a clinical trial for tinnitus and say, I'm going to go here and get my patients to be recruited for a trial. So what we did in our field was we created the Centers of Care network that's advertised by one of our partner organizations, Care PSP. And that alone was enough to get industry buy-in and investors to invest in clinical trials for progressive supranuclear palsy. Because before that network, they assumed that a trial would be impossible to recruit for. So I think that's number one. And then number two, we need the quantitative diagnostics. It is very difficult for a venture capital investor and for a company to buy into a technology that's subjective. There needs to be objectiveness to it. And that is where it's really tested in a clinical trial. So those two things are critical. And then the investors and the companies will come.
Thank you, Amy. Let's go to Winnie.
I just want to say I'm a big fan of the idea of bringing funders together, and I think that's very new to the field, and that's something that really can help us board. And I would like to motivate you to go even one step further, and maybe by next year, we have a meeting, such a meeting maybe again. Why not having also the national funding organizations here on board, like in Germany the DFG, in Switzerland the SNF, and maybe even the EU. And my dream would be that maybe very soon in the future, we will have a call for application for tinnitus research. It's something that has never happened. I've never seen a call for applications just dedicated for tinnitus research by any of the national funding organizations. And of course, they also have lots of money, and joining with them, that could really stimulate research in around the world a lot.
Definitely. Indeed. Marlies.
I have also a point which Amy addressed, and I'm very, very thankful, Amy, that you addressed the problem of the you need a quantitative biomarker. This was your message. So I think I first wanted to say, I assume that we may be much more progressed than we think, because there is already an agreement, at least in 2 points among the whole community. So this is hyperexcitability, and then these alpha-beta change at rest in the cortex. So we analyze tinnitus since 30 years. 30. We started 15 years animal research, and we focused on objective biomarkers. Then we switched since 10 years on humans. And we focused there again, using the objective markers from the tinnitus in the animals to this. We distinguished tinnitus with and without hyperacusis and hyperacusis from the very beginning. We always found the same. We found at least 4 biomarkers which can be quantified on the level of the bearer, RBR wave, fMRI addressed, evoked, EEG, and MEG. The only difference to the community here is that we distinguished tinnitus with and without hyperacusis. So would you give money to the community that they restart to do this? Perhaps they are already much more progressed in this and could speed it up by a very simple subclassification. Perhaps. What is your opinion? And we could confirm with our studies what Dirk de Ridder found, Vinnie Schlee, Nathan Weiss, and all the others, but add a little bit on. So perhaps we are more progressed than we think to have this tool. What is necessary, as you just said, Sven, to speed it up and make it more cheap for the industry. Because if we would have a biomarker.
Something that distinguishes groups of subjects isn't a biomarker necessarily, right?
Sorry?
Something you measure that distinguishes two groups of participants isn't necessarily a biomarker that can be used at an individual level. And then,
Oh yes,
and then
you can do the same. You only separate them. So you only do it on the.
The individual variability within the group is great enough that
No, I presume so someone who followed. So I will not talk about our studies and I will not do it in the presence. But we found, so it's since 10 years. I can only emphasize we repeat the same, and we on different levels. It always. And I just make a test with Dirk de Ridder, and I just.
I just wanted to add that like
so my
doing
point is
group-based comparisons is one thing to consider.
Yes,
Another,
I
like
see
let's say I had a
I
biomarker
see this point with the group base,
Parkinson's
but
disease.
I just discussed if we would have a tool to bring it to the individual level, and I presume that's feasible. So I will not say it. So it's, yeah. Sorry. So my point is only if this would be approach, which is why only 2 questions, nothing else.
And I was wondering, are you speaking about a specific biomarker? Because you're speaking about biomarkers in general, but as being said, there's a lot of variability within those biomarkers. There's not one thing that really characterized. So if you're saying what you just said, is there a specific biomarker you're thinking about?
Yeah, perhaps we have to talk about what is a specific biomarker. So we talk about functional or molecular. So this is of course a difference, and I do not know what we are talking about. So I'm speaking about functional biomarkers. Functional biomarkers, meaning you have a characteristic feature where you can recognize it every lab. Either every lab confirms this or not.
That just doesn't exist. So
yeah, the question is, does it not exist? This is my question.
That's all.
This here is a question.
Yeah.
Does it not exist? Does not everybody and every lab confirm this? Because we do not subclassify. So this is my question, Bertolt, and I do not know if we know the answer. I do not know. I only.
I think I think the subclassification is an important step, but even the subclassification, we have no measurement that has requirements for biomarker at the moment. So I think we have these group differences, but I think the 2 points, we have the high variability and also we have no specificity. So I think the only group who looked on that was Sven and Dirk, who had a good results in separating tinnitus patients from controls based on EEG. But as soon as they integrated data from patients with other pathologies, the whole system collapsed. So that shows that these measurements were not specific.
I think
it's good to define what success would look like if we were to achieve this long sought for biomarker. So taking just the study we published as an example. So we using this videographic measure with the way that the emotionally evocative sounds make the face move, that counts for, was it like basically 76% of the variance of the individual THI scores, right? So where is that's not good enough, but what is the definition of success for an objective biomarker? It would need to be individually predictive, but sensitive and selective, resilient. I mean,
unless we define what it would be, then we all think it might be cabin, right?
Yeah. And the thing is with those biomarkers, we also study them in very selective groups, which is the first thing where you have to study them because they're most related there. Very much the associations are very much higher there. But then if you bring it, as Bertolt said, to other groups, it evaporates. There's nothing left. And the other thing is that I do understand why we would want a biomarker, but I'm also not fully sure what it would solve if we get that one biomarker.
Right. And I want to raise the question too. We have instruments today, the THI, for example, the TFI, for example. They all have subscales associated with them. So when we say biomarkers, what do we mean? Are we relating that to the aggregate score, or relating that to subscales within these instruments to get more specificity?
It's a biomarker is biological. The questionnaire doesn't.
How are we
measuring anything?
How are we going to measure it?
There's a stamped on it.
Oh, so biomarker is biological?
Not necessarily. This depends on your field.
In multiple times.
Yeah, it can be multiple.
How do you measure it? That's key.
One more factor is, so if, say, I've got a biomarker for Parkinson's, I measure the EMG, right? And I can, there it is, objective measurement of the tremor. Okay, that's useless. That's sort of it's measuring the fumes out of the exhaust pipe. It's not really, I can measure it. It's quantifiable. It's highly related to Parkinson's disease. It's measuring something so far downstream of the disease generator that it isn't, you wouldn't use it to target a therapy. It would be too far downstream. Or say I'm interested in stroke and I'm looking at the diameter of the aorta. Well, it's true that stenosis of the aorta is a predictor of stroke, but if you really want to measure stroke, in the brain, you'd want to use a vascular measure in the brain. So I think what you're measuring as far as whether it's downstream or upstream of the purported tinnitus generating mechanism is also a really important consideration. So genetics, right? Genetic predispositions, which based on the studies I've seen account for somewhere between like 0.5 and 1.1% of the variability in what's being measured is maybe very far upstream for an acquired disorder. And perhaps the example I gave of sort of emotionally evocative reactions are downstream. We wouldn't want to treat that. So what is the right point on the spectrum?
I'm not 100% sure I agree that they're not useful when they're downstream. I had the pleasure of being in a clinical trial where they were using a tremor biomarker, and they were showing us in the real time while the person was in the high frequency ultrasound machine, the biomarker went down as the physical tremor went down and the person couldn't fight when they came out. I think those types of quantifiable endpoints are more important to companies, wanting to invest in a technology. So even though it may not be the root cause in Parkinson's, the resolution of the symptom is measurable. And that is also an important biomarker to consider.
Yeah,
Measure even.
To validate for treatment, then that is a secondary clinical endpoint. It's valuable. As if it's meant to identify a possible target for treatment, then I'd say that was what my comment was really.
But you could also have, let's say, the tremor that you're measuring at the end. You could implement that for improving a treatment, for example, by closing a loop, for adjusting whatever stimulator they put in.
Yeah.
Then adjust it automatically rather than visually. So I do think that those measurements anything which is quantifiable can probably benefit a treatment approach if you use it for closing a loop.
If you use it for closing a loop, but in many instances, it will be just one part of the issue and not the full thing. One biomarker is never, or almost never, the one thing that is the solution to that is the treatment solution. So it can be, but it's not the only thing that we need to focus on. And it's one of the things.
This is the spike protein of SARS-CoV-2.
Yeah. Well, in truth, you all will need many different kinds of biomarkers. And I think that's what this conversation is coming to, is I only mentioned the clinical trial progress biomarkers to see even small changes in someone's tinnitus in the clinical trial that's quantifiable. As somebody mentioned also, this doesn't tell us anything about mechanism. there's a whole other field of biomarkers that are basic research biomarkers that will have no clinical use whatsoever, but they're helpful for basic research. You all need all of those.
I think the example of the Parkinson tremor is very good because when anybody wanted to illustrate how well deep brain stimulation works for Parkinson, he demonstrated a video with deep brain stimulation on and off, and the huge tremor just stopped, and patients could move normally, and then again they had a huge tremor. And this is convincing for everybody. But for sure, we agree that we need much more outcome measurements. We need to find out what is the long-term effect, what is the quality of life of these patients. So the tremor is not all their problem, but it's very helpful in order to demonstrate the efficacy of a certain treatment. So I think we have to be aware that each biomarker has its limitations, but it's, I think, very helpful also to convince somebody or to demonstrate is something working or not.
We seem to have lost our chair, but if I can ask a question coming from an outside field. So I've worked in the pain field for many years, and in the pain field, there is absolutely no biomarker. The only thing that people have found useful, and everybody knows it's an absolute rubbish measure, but hey, what can you do, is to ask them, how is your pain, or a scale of 1 to 10? And they give them a putative analgesic, and they ask them, has your scale of 1 to 10 changed? And they well, it was 6 before, and now it's 4. This is not a biomarker, but the fact that and there is no biomarker at all, this has not prevented progress in the field. In other words, many analgesics have been developed and have gone out into the world to treat people with nothing more than this scale of 1 to 10. So aren't we being a bit pessimistic to say there's no biomarker? You can ask people, where is your tinnitus on a scale of 1 to 10?
But that depends on what you find a biomarker. So I just heard that some people, in some fields is a biological thing. In diagnostic accuracy, it can be also skill or whatever. So it depends on what you find that is what you think that is a biomarker.
Yeah, for me, a biomarker is something biological, like the concentration of tau protein.
Yeah,
that's a biomarker.
Yeah,
It can be behavioral as well. It can be behavioral, a biomarker.
Yes, for me it can also be a
It's very broad.
Yeah,
very broad concept.
Can I just add from the patient field? So why did we ultimately, use our slogan is to basically silence? Of course, everybody knows what that means, but if you would ask 100 patients across the globe, different backgrounds, probably different tinnitus subtypes, then you would get the answer that 2 things about their tinnitus are the most important, where they consider their quality of life to be better. One is the word loudness. Whatever loudness means. Of course, as we all know, for some people, loudness is something different than for others. But even though it's something that a patient, I would say, can predict quite well for himself. So I always said, if you would offer tomorrow any patient in the world the chance to lower his tinnitus loudness by 50%, and that's it for the rest of your life, I'll tell you 95% of all the patients in the world sign that contract and are happy. That is one thing. The other thing that I think is underestimated is frequency. So as we all know personally, lower frequencies are something we can tolerate much better than high frequencies. So those are the 2 things I just want to mention from that side. And the third thing I just want to say about biomarkers is, I think what's very hard in the field, and this is why I'm a little bit just by feeling on what you say, Mr. McDonald, is the situation that we try to find a biomarker that tells us what. So tells us that the one biological factor has decreased through a treatment. But the problem is that we're taking out of the account is that there are people who consider themselves, and this is also very hard, that they have a severe tinnitus. Like I would say I have a loud tinnitus, whatever severe means, but I have a loud tinnitus. But I'm super well adapted to it, and it's something that does not prevent me from having a normal life and do whatever I want. But there are people that have a considerably low tinnitus, but they're impacted extremely. So if we find a treatment that lowers the score for them and me, what is the outcome measure of that? And this is something I till today, maybe I'm too stupid for that, but that is something I really have problem bringing together in this field as an outcome measure.
I want to echo, Sven, what you said about loudness, because during the last 8 months or so, my group has done a requirements gathering where we queried many of the major tinnitus support groups in the United States. Far and away, and this was not a scientific study, but far and away, loudness was the thing that patients wanted to see addressed most.
Yes, number one.
Number one. By far, I will say by far.
Yeah.
Yep.
And so, I think the other reason why biomarkers are important, obviously, is when clinical trials don't work, and you want to know, has there been a biological kind of effect? So I think some of the trials in the past, you don't get the kind of like the outcome on the tinnitus affected, but did the target get engaged? Like, was there a biological effect? And so I think that's important. And so I was just listening to this conversation. I wonder if a potential sort of action for this group is to actually, picking up what you were saying, Dan, sort of actually define what do we mean as a field, as a group, by biomarker, and define that. And maybe there are different types of biomarker. I think it'd be helpful to go through the process thinking about and agreeing how would we use this biomarker, and why is it important? And perhaps if there's consensus around the types of biomarkers we need, why they're important, how the field will use them, then at least we can then sort of start to Marlise's point, agree whether we already have that, and if not, as funders, we can target research at delivering something very specific that we know the field needs.
Perhaps I can briefly add. So if I got the point, would this not be an in-between step? If we would agree, I do not allow myself now again to say functional biomarker, but let's say characteristic features of tinnitus, independent of hyperacusis or not. If we would agree to something like this, and we could reverse with a therapy 1, 2, or 3, these kind of features would be then not be a step further, and from there specify and look what is behind this characteristic feature? It's a question. This means my point is only perhaps in between we could live with group levels and could do and look, can we reverse it on the group level without having this specific on the single level? If we would have an agreement, this is something, and we would learn if we can reverse it with a therapy 1, 2, 3, then we would learn, this has something to do with tinnitus, or this has something to do with hyperacusis. Would this not be a step in between?
So I think the biomarker has to work on the individual level, otherwise it's worthless. So the change of the biomarker has to correspond to the subjective change of sensation. But I want to come back to the comment of Sven. You say loudness is most important, and next sentence you say whatever loudness means, and that's also like because if you measure loudness with a VAS scale, like in pain, you have a completely different result compared to a loudness match, and so that's a difficulty. And even you would say, for sure, I would feel immediately if the loudness decreases by 50%. Yes, if it decreases now. But if I ask you, how is your loudness now compared to 1 year ago?
I can tell you.
You can't tell.
Yeah.
So that's a because you have it. So that's a difficulty that I understand exactly the request from the patients, but how can we operationalize this?
I'm 100% on your side. I'm just maybe I said it the wrong way. I'm just saying we have to prepare all of us for this situation. That we, for example, find a biomarker, let's say for loudness, whatever that means. We just have to prepare ourselves that we find a good biomarker, and it said, well, after the treatment, this biomarker shows your loudness should have decreased by 50%.
Yeah.
But the patient will not be 50% better. This is, I'm just saying, the one thing we have to prepare. And then probably there's a secondary look on how, because maybe I'm wrong, but the tremor example is a very good one. So if somebody's got a tremor and holds a glass of water, you can measure how strongly he shakes, how he grabs it. And he will probably tell you that if this is 50% better, that he will feel 50% better. This is just my hypothesis. In tinnitus, that will be different. So the question will always be, what are we measuring and what will be the output for the patient? And this is something that I understand makes this field so super complicated. So I'm just really putting that.
In pain research, usually what is measured is painfulness with a numeric rating scale. Some kind of suffering questionnaires, which are different, like pain catastrophizing, and disability. Now, what's very interesting is that with certain spinal cord stimulators, you can improve painfulness, disability, and suffering better than with another stimulation design. I mean, quantitatively, a lot better. And then you ask another question, which is the PGIC, the Patient Global Impression of Change, is no difference.
Yeah.
Even though with one stimulation design, or in this case it was a closed loop approach, every measurement was better for questionnaires, for numeric rating scales, but the patient global impression of change was the same. So they didn't really care whether they were better with one than the other. So there has to be something else. Could the cost benefit analysis would say, well, this gives better outcome, but it comes at too much of a cost. But the point I wanted to make is that, to come back to Sven's point, is that the patient's global impression might actually not always be reflected by the outcome measures that we use. And this is a problem.
Yeah. Jeffrey had a question.
I'd like to make
Bertolt, I
two
think.
questions. One is I have type 2 diabetes, so when I get home, my girlfriend's going to give me a continuous glucose monitoring device. So the question I would raise is, as a transient feature of tinnitus, in terms of measurement, these measurements are time-based too. They can be ecologically based, very real-time. They could be yearly, clinical appointments. So this introduces a whole level of complexity in terms of how do we measure improvement, right? The other question I would ask is, with respect to the data sources that we have available, I'm thinking mainly of the Unity data source, how much has been mined in terms of the subscales within the questionnaires and other data that was I'm not mined it myself. I have access to the metadata, but the question maybe for you, Bertolt and Winnie, how much has been mined with respect to the subscales and the impact of trying to define not biomarkers, but cognitive behavior biomarkers, if you will, to define tinnitus? Does that make sense?
So I
I don't know who can answer that question.
Forward the question to Vinny, who is in charge of all the data management, but we will make all the data available soon for everybody to that. I think I wanted to comment on Dan's comment. I think it's a very important problem, because this might happen that we improve for somebody concentration and sleep, but the patient is still not satisfied, and the satisfaction depends on expectations. So if the expectation of the patient is, I want that my tinnitus is gone, so an improvement of sleep and concentration might not satisfy. He's still suffering. And for sure, all these successful therapies have in common that they do some kind of expectation management. Let's say TRT, CPT at the very beginning say, we won't change anything about your tinnitus sound, but we will help you to deal better with it. So then you have a higher chance that patients are satisfied at the end, because you manage the expectations. But I think that's also a very important part about this. What really makes a difference for the patient, what is the expectation of the patient?
Couldn't agree more. On the clinical side, in the learning health network model that we're building, expectation management will be built into the algorithm, right? It's part of it. It has to be.
But just to manage, let's say, resources. I mean, this is also a question is, where we don't have an unlimited amount of resources. So we have to define what are we targeting. This is what I'm just saying is you're absolutely right in terms of how complex it is, that how somebody perceives the tinnitus and how he's debilitated by it. But we're just saying, if you want a success for the majority of the patients, and I'm sure there are in the end some subtypes that are more complex and have comorbidities and so on. But I can just tell you from speaking to many patients, and I think Hazel would agree, and speaking with even more, is that loudness is one of the main factors that if you would reduce the loudness of a patient, 90% of the patients would give you a positive response. I'm pretty sure about that. I know that's not covering everything that we need, but I'm just saying in terms of managing resources, I don't know if that's something to look on, but I can just tell you that from the reality of people we're speaking.
Yeah. I mean, patients cannot objectively measure their loudness level. They can't.
No.
Yeah, I want to hear from Dan, because this is one of the points that Dan brought up. Loudness matching is not an objective measure, etc. So
I understand.
Yes, it is the most, that would be the heart, the seed of the tinnitus distress, if you could measure the loudness of the phantom percept. But it is, I think what we're trying to say is it's, sorry for my American crassness, it's sort of fucked as a biomarker.
Absolutely, yeah.
It's unsolvable because, I mean, anytime you try to estimate tinnitus properties with an acoustic signal, is a question of whether the internally driven percept is really relatable to the sound-driven activity. So for acoustic loudness matching, if you have hyperacusis, which many people with tinnitus do, you're very sensitive to the level of sound. And perhaps that's why, tinnitus loudness is usually rated at like 10 dB SL. But let's say the manipulation made you more sensitive to sound. Say by virtue of having listened to some sound therapy for 2 hours, you're now hypersensitive to sound. And so now, it might look like your tinnitus match has improved, right? Because now my tinnitus is 10 dB softer than it was before because I'm so sensitive to sound that I'm saying that I'm like, it's really a product of it being hypersensitized. Or say, it's the opposite, right? That your hyperacusis got better, but your tinnitus didn't. It would still show up as your tinnitus got much worse because now your matched level is 20 dB lower than it was. So it is inextricably linked to the perception of sound, which is, more related to hyperacusis. But apart from that, this loudness is a psychological construct. It's different than measuring that sort of detection threshold. So then, right. I do completely, as somebody who has tinnitus, relate to, I just want the loudness to go away. But there has to be some education going backwards too. If we could, we would. This is not like everybody's been thinking about loudness, right? But you can't. You could measure like the total neural recruitment function. Maybe that is like closely related to the perceived loudness. In mice it is. So, maybe that is a closer thing, but again, it's like a sound evoked measure. There is no measure.
And the one thing I wanted to point out too is that in our study, there's a high comorbidity of hyperacusis too, to your point, Dan, that raised sensitivity to sound.
No,
It gave rise to people saying loudness is the biggest issue.
we once did a study that correlated brain activity to a simple numeric rating scale like ease and pain, and we found some correlates between the salience network and the auditory cortex, and for loudness matching, 0. So we couldn't find any correlation with whatever brain activity to a so-called more objective measure of loudness matching, which suggests that, as Dan says, that it's not a measure, it's not an objective measure for tinnitus, at least certainly not related to any brain activity, right?
But from a methodological standpoint, I'm not sure if the objectiveness of the measure, at least if we use it as an outcome measure, is a real issue. I mean, loudness is a perceived outcome. That is, there will be differences between how all of us perceive loudness as disturbing or how loud we think something is. But that using that as a biomarker might be an issue, but using that as an outcome of clinical studies is not an issue, because the thing is that it is about how the patient experiences the outcome, loudness.
Yes, but there is no correlation between the subjectively perceived loudness and the loudness matching. So they don't correlate.
But then use the subjective one, because why use the objective? Because
that's the point.
yeah, nothing is going to change for the patient if we change the objective measure, but they still have tinnitus. So they still have the burden of tinnitus. So
No, but the problem is if that the so-called more objective marker of loudness matching, if it doesn't correlate with any subjective scale for the patient, then we should not use it. The problem is that if you go to, let's say, the NIH or another, they say, yeah, you need something more objective, so you need this loudness matching, which doesn't mean anything. For example, if you have a study that uses it, let's say Susan Shore's study, loudness matching was used, but then the question is, does it correlate with any subjective improvement for the patient? We don't know. But we do know that in general it doesn't correlate.
It's still useful.
and I
think it's still useful, the loudness matching. So it's not trivial to assess it, but it's still useful because so if you have a given treatment, so then you can have an effect on the tinnitus loudness, and it's already something. So it's a biomarker. So, and I think it's Sven points. So what is different? There is a disconnection between the loudness and the existence of the impact, the distress, actually. And indeed, so you can reduce the tinnitus loudness, so you can try to assess it with loudness matching and so on. So you may decrease it by 30%. Without any impact, with or without any impact. So it's really disconnected, but it's still interesting to prove, to show that there is an impact on tinnitus loudness. And then the impact is something else. It's for me, it's another dimension.
So given that perceived loudness is probably the number one thing that patients talk about, how do we work backwards from the clinical side to the research side? If that's the number one thing that concerns them, how do we then define what loudness really is? Because we know patients really can't define it. How can we reverse engineer loudness from the patient perspective to more directed research to address it?
But I'm sorry, but I don't agree that they don't know, because you can just ask them. So that's what. So you probably heard about patient-reported outcome measurements from the PROMIS system, for example, and you can just ask them and use that as an outcome. That is what we're already doing, and that's actually. So you spoke about regulations in the Netherlands. That's what we should be doing. That's actually mandatory to take these outcomes as your most important outcomes. So it's not that challenging, actually. To be honest.
So if I can jump in, I think that there's been a lot of discussion about the biomarker, but we haven't heard three characteristics to me that are important. One is probably the most important is the causality aspect, because for example, if I take Dan's example with the face and the emotional characteristics that are correlated to tinnitus, right? But can you act upon that, and is it actually a consequence or a cause of the presence of tinnitus? So that's extremely important. And then a biomarker should be something you can act upon, right? So if you measure EEG, global, alpha, or whatever changes in the brain, but you have no means to act upon it, I don't think it's a useful, very useful marker at this stage. And there is a difference, I think, with loudness being used as an outcome measure, so that the biomarker is not necessarily can be distinct from the outcome measurement, right? So you have outcome measure, which loudness should likely be still used because it's the main concern of patients. But then you can use also other markers, not necessarily related, but yeah.
Let's cover Marlies and Bertolt, and then let's break for lunch because I'm very hungry. All right. Marlies?
Oh, okay. So I only want to say that when you have, you could also solve this problem perhaps by a prediction. If it's true that, for example, the lower the loudness, And the more the suffering, it may correlate with an increase in the hyperacusis score or uncomfortable score. And either it's right or it's wrong. And the team could test it. Everybody could test this. So we have some evidence for this. I only wanted to say it's testable. We can test this.
The VAS scores for loudness, they correlate quite well with the tinnitus questionnaires, and they also correlate with the clinical global impression. So if patient has a change on the VAS score, typically also the patient tells that the tinnitus improved. I think we have some measurement which is reliable and which can be used as an outcome measurement, and probably that's one of the best things we have. In contrast, I don't agree with you, Arno. For sure, we can also do the loudness match, but we have no clue what it means. It's not correlated with any brain activity. It's not correlated with any other measurement. It has a very poor retest reliability. So we also can ask the patient for the phone number. It holds some information, but it probably does not relate to his or her tinnitus. So that's and concerning the facial expression, it's very easy to act on facial expression. Just put Botox in the face, or probably it will change the tinnitus. Good try.
Just to answer your points. So I guess one thing against the VAS and so on, but it's composite measure.
Yeah,
And
sure.
it's the whole point.
But
So
it works.
when you ask, it's not trivial. The loudness. From low level to low percept to high percept. The thing is, so we try to be accurate. Okay, so it's one way to assess if it's possible for tinnitus, so the loudness. But the thing is, maybe we mix with the VAS. On one side the VAS, the loudness, but on the other side, what could be maybe the volume, the space tinnitus takes in the consciousness.
Yeah.
So you can have something. We were discussing about mosquitoes yesterday night after a few years.
That's a
But
good one.
it's very small, but it's very bothersome.
Yeah.
And then so the entire consciousness is focused on the mosquito, and so it's very small and it's super big in terms of consciousness, and it's the problem with tinnitus. So it can be super small and big in terms of the impacts related to the consciousness.
I agree completely.
Yeah.
I think that subjective perceived loudness is kind of reflected by the connection between the auditory cortex and the consciousness network, the salience network for sure, and a stress-related network. So all these interactions play a role for the subjectively perceived loudness.
And we looked at that in pain. So we gave patients with pain before and after a treatment a numeric rating scale for painfulness, suffering, and disability questionnaires. And what was interesting, and then we did the cluster analysis and we found 3 groups. One group had a painfulness of less than 5. Suffering questionnaires were normal, disability questionnaires were normal. The second group had a numeric rating scale of 6 with abnormal suffering, no disability. So they were functioning, but they were suffering. And the third group had a numeric rating scale of 7 for painfulness, abnormal suffering, and disability. Now, if you look at it from another point of view, from a treatment point of view, the first thing that should improve is disability. If you just decrease the loudness score by 1, if you say start at 8, you drop it below 7, disability will improve, but you have to drop it below 5 for the suffering to improve. These are averages. So there is a correlation between loudness, at least in pain, between loudness, suffering, and disability, but there might be 2 thresholds. And depending on how much you can bring it down, you can improve only disability or only and suffering, but the loudness the painfulness is critically important, just like loudness is, based on what Sven mentioned earlier on.
It's nonlinear, so it's nonlinear effects.
Yes, it's a nonlinear
And it's
effect.
also the difference between pain and tinnitus. Pain, it's really the alert signal. So there is a correlation. So between the loudness and the problem, the severity and the problem. And for tinnitus, it's not that clear. You can have tinnitus and you can do well with it.
Yeah,
So
but
it's not exactly the same meaning.
the point I make, don't get me wrong, it's not that I say, okay, it would be ideal to correlate a biomarker with loudness. I'm just saying is where, in terms of resource efficiency, is where do we start to look for something? It's like asking you go to the jungle and find the tiger. If you haven't seen a tiger, you'll probably bring home a hippo and you present a hippo. So this is the I would say just the thing I would like
I
to.
think the loudest biomarker is the most important starting point, because how will you otherwise know if you're going in the right direction?
But I'm just saying, I think from a patient view, as all of you who saw patients, you will see probably two groups of patients. One patient, they will tell you, my tinnitus is always the same. It's in the morning, in the evening, if I have bad days, good days, it's always the same. And then you will have a quite large, I would say, proportion of your patients, like I am, that you'll see me 10 days in a row and I give you 10 different answers, because the day is different. Yeah. The day is different, and it's not even, and that's the problem. We often, like in patients, we often talk about stress. Stress increases my tinnitus. Well, yes, but there are probably 5 million different factors. I can identify myself. I can tell you, it's a day. I had a perfect sleep. I woke up. I don't know. I did some exercise, had a nice orange juice, and the day is fucked up. Why? I have no idea. So this is the point, is where, how can we measure this group, which is, from my perspective, but as I say, I'm not an expert, that's almost impossible. That is almost impossible. And that is a large portion of your patients is that way. Absolutely. The ones that have one stable tone, it's perfect. That if that would be the case, it's perfect, because it's right away they can tell you. Of course, relatively, I can tell you that the shit days have become better. that is something, of course, that every patient can tell you. But I'm just saying, this is a quite large portion of the patient group that makes it a little more complex, I would say.
Yeah, and if patients by and large complain about loudness as the number one distress, from a PR perspective, how do we tell them, well, you don't know how to measure your tinnitus, you don't know how to measure your loudness, we don't believe what you're telling us, you know?
Oh, we have no idea what...
Yeah, we can't tell you what loudness is. You're telling us you feel loudness, but we can't tell you what loudness is.
Yeah, no, that's a problem.
Right?
Guys, let's break for lunch.